All right, so first question. Which of these is the production of new blood cells? It is hematopoiesis. Hematopoiesis, too many vowels. So it's A. Which of these cell types breaks down bone? It's osteoclast. Does anyone know the trick to remember the two? Yeah, blast, build, class, carve out. Exactly, that's kind of the trick to remember those two. So the ones that break it down are the osteoclasts with the C. Osteoblasts build it up. Osteocytes kind of maintain it. Not part of bone ECM, water, collagen, mineral salts, or fibronectin. So fibronectin is not. Fibronectin's for blood clotting. Not a zone of an epiphyseal plate. Resting cartilage, hyaline cartilage, hypertrophic, or calcified. Hyaline cartilage. And which of these is a situation of bone formation? Growth of bones during infancy, repair of fractures, remodeling of bone, or all of the above? All of the above. Very good. Nice work. All right, before we start the chapter, is anybody here who didn't take their test yet? You want to step outside for a second? I'm just going to go over a few questions that a lot of people missed. Or you can just stay in here and we'll give you all the answers.
Just hide in the corner. I won't let you do
We're going to go through all the questions, but I picked out a few that a lot of people missed to look at. So first up is Tim here. And what's cool about this is you can actually see what people wrote for these short answer questions, which is where a lot of people missed points. But this way you could see like what a perfect answer would look like. Maybe compare it to yours. Obviously, we don't know. Don't shout out which one's yours. I don't know whose is whose. So this one was difference between negative and positive feedback. Let's find one that's pretty good. So this one was almost, so it's giving an example. So it's almost perfect because positive was, the example we used with the baby being born. Negative was in high blood pressure. Let's see if we can find one. All right, so this one's pretty good. In negative feedback, there's a decrease of the stimulus. So you remember when the body was doing the negative feedback, the response got rid of the stimulus. Positive is different. So I would have said positive increases the stimulus for that one, just to be specific. Oh, so here's one. Negative feedback decreases the stimulus, while positive feedback increases the stimulus. So that would be like a perfect answer there for that one. Same with this one. And if you remember, did you have something on the study guide that looked similar to that question? Was it identical to that question? So that's not a mistake. If it's kind of a harder question like that, I'll put it on the study guide, and then I'll just put it on there basically word for word. Because it's something you had to think about beforehand and figure out. What other questions? 17. Seems like the button went away for me to click on it and see them. Maybe if I refresh it. Weird. Okay, anyway, so the differences between mitosis and meiosis. In order to get full credit, you had to actually describe two differences, saying that mitosis happens everywhere in the body and meiosis happens in the testes. How many differences is that? That's one difference. So for another difference, you could have said meiosis makes sperm or eggs, mitosis makes all the other cells. Would have been another difference. Another thing that a lot of people were confused on is they said that meiosis was fertilization, and it's not. Remember, meiosis is only making the sperm and the eggs. It has nothing to do with the actual fertilization process. Fertilization is its own thing. So meiosis happens, and then fertilization happens. So those are some common notes on that one. Let's see, 36. Most people did pretty well on the function and location of the different epitheliums and fiber cartilages. Similarity and differences between epithelial and connective, that was a hard one for people. So the similarity could have been they both make up serous membranes. Would have been one similarity. Some differences. Connective has blood vessels, epithelial doesn't. Epithelial has lots of ECM. Epithelial has very little. Epithelial has lots and lots of cells close together. Connective, the cells are really spread apart. So I'd have been like 3 or 4 differences there for that one. And once again, was that one on the study guide? So just word for word on there. So you want to make sure, for future tests, if there is something kind of long or seems like it's hard on the study guide, make sure you do take some time to know it, because it'll probably be on there. And I did make that 36 question bonus because it was on a group assignment, but I think we never finished it or went over it, I believe. So I made that one bonus, and then I gave you all a few other points back too for questions that seemed a little too hard or a lot of people missed them. So altogether you got about 10 points added onto your grade, which you saw in there. And of course, if you ever want to look over your specific test, you can come any office hours, and I'll just give you the computer. You can just look through and see exactly which things you missed. The reason I don't just give you access to the whole thing is because I'm going to use those results to change how I teach certain things for the future. So for example, if there's a certain question that a lot of people missed, I'll go back and change how I taught that and see if it works better. But if I give you access to the test, I can't use the test again. I have to make new questions, and then I can't really tell how things are being improved. But that doesn't mean you can't come look at it anytime you want. Look at each question, see how many points you got, all that business. Any particular questions? Okay, so chapter 6 is all about bone tissue. So like the microscopic structures of bones, how they form. And what kind of tissue is bone? It is osseous, and in terms of category, it is connective tissue. So that means there's going to be types of fibroblasts in there. There's lots of extracellular matrix. So you'll see some similarities. So we're going to start with talking about how we make bone from the beginning. And so when you're an embryo, you start to make your first few bones. We've got some basically stem cells called osteoprogenitor cells. They turn into osteoblasts. Those osteoblasts secrete collagen, mineral salts, start making the bone. Once the bone's made, they turn into osteocytes. So that's their final cell type they turn into. These cells secrete a small amount of ECM and keep the bones healthy and alive, basically. And then a separate kind of cell is that osteoclast, and that's the cell that breaks down the bone, which we'll get into later. It seems a little strange that you have a cell that breaks down your bones, doesn't it? We'll talk about more later, but the reason is if you break down your bone and rebuild it, it keeps it really strong. If you just made your bones before you were born and just kept them that way, they'd get brittle and break really easily. So these osteoclasts basically recycle the bone and keep it fresh and strong and young. All right, so pretty cool. Your bones when you were first developing into a fetus, they just started as mesenchyme, which you remember is embryonic connective tissue. So it's just sort of a pile of cells in the shape of a bone. It's not hard yet. It's not really cartilage yet. It's just kind of some bones in that shape, or some cells in that shape. They do keep growing till 25, at least a little bit. And that word ossification, which might be on your study guide, we'll see it a lot. It's the idea of building bone, but not from scratch, building bone on top of other tissue types. So either replacing the mesenchyme with bone, or as you'll see later, replacing cartilage with bone. Those would both be ossifications. All right, so the hardest thing in this chapter is these two, and that's how we build bone. So there's two different ways. We could do it with intramembranous ossification, and that's the process the body uses for your flat bones, which are in your skull, mostly your facial bones. And then endochondral ossification is the other type that some bones are made. And so we'll look at an example of a long bone. So all the long bones in your body, your arms, your legs, your fingers, your toes, they were made with endochondral ossification. So I'm going to give you some simplified steps for each one, then we'll watch a little movie, and then in a little bit you're going to do an assignment with those. So intramembranous, we start with osteoblast secrete collagen. So that's, part of the bone ECM, but it's not the really hard part. They change into osteocytes, deposit mineral salts. Those mineral salts make columns of bone. They fuse together to make trabecula, and then the edges turn into compact bone. And you can see that in here. So we started with mesenchyme, so just like those cells in the shape of a bone. Then our osteoblasts secreted some collagen here, which is like the blue strands. Then they got the calcium mineral salts deposited in yellow there. And they turn into osteocytes. Then these little pieces of bone we made fuse together to make these trabecula, which are in yellow. And then the edges, or the outsides of the bones, become compact tissue. So you've got spongy bone in the middle with holes in it, and then those trabecula going through, and then on the edges, you've got that hard, compact bone.
Are the trabecula hard?
Yes.
Or just the solid pieces that go through the spongy
Right.
bone?
So these trabecula are just as hard. It's just they're narrow columns, whereas this is hard and completely solid with no spaces. So we've got a sandwich: compact, spongy, compact. So if you're going, looking at a skull bone, if you like drilled in, it'd be compact, spongy, and then the part facing your brain would be compact too. What?
I kind of want to do that.
All right. Computer. So we'll look at a little movie. For 45 seconds. All right, so we can kind of see it developing. Mesenchyme, those osteoblasts secrete collagen out here. And then they secrete those calcified salts, which are like rocks, basically. They're like hard minerals. They turn into osteocytes. And then those pieces of bone that we're making, those small little places, start fusing together, which just happens like so. And now we've got a whole trabecula. So we've got little places where you start to make the bone, they fuse together, make trabecula, and then the compact bone forms on each side. And just as a slightly more zoomed out picture of the bone that you ended up making with the compact bone, spongy bone sandwich. Oops. Right. Any questions on intramembranous ossification? Besides that word's really big.
When the osteoblasts turn to osteocytes, does that just happen just as they mature, like as they go through like so many cycles of secreting collagen, they just kind of stop and
Yeah. So it's actually they sense when they're surrounded by the calcium salts, and then they turn into osteocytes. So they can tell, all right, we built up enough bone, let's go into osteocyte mode. We don't need to keep building the bone larger.
that's just when they just do a little bit, just enough to like maintain.
Right. And I do have a worksheet for you to practice with these because I know it's a lot of steps and it's a little confusing. So I'm going to give that in a moment. So our second kind, this is to make our long bones. You may see some similarities and some differences, hopefully. So we start with mesenchyme cells. And what's different about this one is those cells turn into chondrocytes. Chondrocytes make cartilage. Kind of sounds the same, right? Chondrocyte, cartilage. So these the cells that make cartilage. So the first thing you do is turn your mesenchyme into cartilage. And then the osteoblasts come in, deposit collagen, mineral salts, and replace the cartilage with basically bone materials, the collagen and the mineral salts. These steps repeat over and over again, traveling through the bone. Like it'll start in the middle of that bone or that clump of tissue, spread outward, and make trabecula, just like in the spongy bone. Form compact bone on the edges, and then the last step is osteoclasts come in and completely remove the trabecula that's in the middle. So you end up with, we'll go back to these, but we made trabecula in the middle. This could be your femur or something. And the osteoclast completely broke down all that spongy bone that was in the middle of the bone. And you'll see later, this is where we can fill that up with red bone marrow. That's what the body will eventually do. And surprisingly enough, that bone in the shape is strong enough to, support all your weight and everything you do to it, even though it's hollow in the middle. It's still way, way strong enough because the bone material itself is so strong. You don't need it to be like a solid bone. And then that would end up being way too heavy anyways. All right, so after we remove the trabecula, we've made most of the bone. But what's so strange about our long bones is they're actually made in 3 places. So you make the middle part of the bone, and after you've made most of that, then you make the ends of the bone, like the parts that'll go into the joints, basically. And this part actually happens after birth, which is kind of interesting. So when that baby's born, it only has real bone in the middle of the bone, and up on those top parts is just kind of cartilage. It takes quite a while. I don't know about that one. I believe you, but.
I know, yeah, I know, little, babies don't have kneecaps, and I don't think they have kneecaps until they're 3 or 4 years old.
Yeah.
Yeah, that's why you can't break babies. That's why they're made the way they are, to withstand new parents.
Yeah, well, withstand the fact that they don't have any balance or anything.
Fall down
But after maybe a year old or so, most of this process is finished. But you actually have a gap between the long part and the ends that's still cartilage till you're in your 20s. And when we dissect our chicken bones next week, we'll cut into them. You'll be able to see that there's this spongy cartilage between the ends and the middle of the bone, which is kind of crazy.
And that's the epiphyseal?
Yeah. And we'll talk about that more later, but it's kind of crazy. All right, so this happens at the ends. Very similar to what was happening in the middle of the bone. So mesenchyme turned into chondrocytes. They make hyaline cartilage. Osteoblasts deposit bone ECM. So if you notice, this is all the same as the middle of the bone. Step 2 and 3. Keep going until you reach the edge. And that cartilage remains between the end of the bone and the middle.
Is there something that relates the name to what it does? Is endochondral inside, chondrocytes or something? How does that relate to what the process is?
Well, the chondral part tells you that it's chondrocytes, and chondrocytes make cartilage.
Intramembranous? Does that need anything really?
I am not sure why they called it that. I wish there was an easy way.
What exactly does chondro mean?
So chondral is a prefix that kind of means cartilage. So endochondral means it involves cartilage. Chondrocyte, cyte means cell. So a chondrocyte is a cell that makes cartilage. So yeah, it's good to break those words down. Mm-hmm. Definitely. And these will be some that you'll probably have to look back at, watch some movies on your own to really get them, because I know it's a lot of steps. And if you notice, is there any osteoclasts in this process? So the ends of your bones stay spongy. You make your trabecula, but you don't get rid of them. And so you end up with something like this. You've got your hollow long part, and then the top of the bone you just made into spongy bone. And then right here is that line of hyaline cartilage between the top of the bone and the middle of the bone. And we'll look at later, this is where the bones actually grow from. because you get to this point probably by about a year of age. But, I mean, your bones still need to get, 5 times longer from when you're a year old to when you're an adult? So they grow from here and grow downward, which we'll see that a little later. So just an overview. Started with mesenchyme, built some cartilage in the same shape. So you start with a little cartilage bone right there. Then we'll replace the cartilage with bone, like the collagen. Erase the cartilage with collagen and mineral salts. Those bones will fuse, and then we carve out the middle, and then form the compact bone on the ends. Right. And then the secondary part we've been talking about starts up at the ends of the bones, in a very similar process, except no osteoclast at the end. You just create the cartilage, osteoblasts lay down the collagen, mineral salts, and then you're done. Form the compact bone on the edge, and then you're all done.
And like that hollow part and the diaphysis is where the red yellow marrow is?
Yeah, that'll be the yellow marrow, and we'll talk more about those in a little bit. All right. And hopefully next week we'll look at a chicken leg bone, and you'll be able to see most of these parts. So see an artery in there, see the epiphysis, diaphysis, all that business. Right. And may not be obvious, but everything that's happening up here at the top, where you had the epiphyseal plate, you've got one down here too. So it's all more or less the same.
So do both of those stay as cartilage, or is only one of them?
Both of them. So you're growing from like both sides.
And you have the epiphyseal plates in all of your long bones, right?
Right. And then after you're 25 or so, they fuse together, and you end up with like a line there of like hard bone. All right, so I'll let you, if you want to watch a movie on that a few times to help you, probably a good idea. Let's see how you could do on this one. So I'll have you do it on your own first. And I've got 7 statements. You have to tell me whether it's endochondral, intramembranous, or both. Right, so why don't you compare with your group, and I'll tell you there is 3 that are both, 3 that are endo, 1 that's intra. All right, so let's go ahead and go over those. Hopefully after you've done this and get the answers, it will clear things up a little bit. So the first one, osteoblasts secrete collagen. That is both. Happens in both. That's the basic way you make any bone. That'd be like one of the first steps. Forms long bones. That's endochondral. Starts with mesenchyme. That's both. All your bones start as mesenchyme, which is that just sort of cells in the shape of a bone kind of thing. Osteoclast break down trabecula in the center. That's endo. Trabeculiform. It's both. And forms flat bones. That's intramembranous. And cartilage is replaced with bone tissue. That's endo. So one thing that'll help you a little bit. So in intramembranous, it goes from mesenchyme to bone, more or less directly. But in the endochondral, it goes mesenchyme, cartilage, bone. So there's like that extra sort of step in the middle. And so probably the best way to study for that part is to come up with your own 2 or 3 sentences for each one. That breaks it down, makes it simple, but it's your sentences you came up with. It's a lot easier than trying to memorize, paragraphs from the book or something. Definitely you can get it down to 2 or 3 sentences without a problem, I think. Just keep it simple on those. All right, so as we mentioned, at that growth plate is where the bone's growing. And so as, once you're born, from the whole time till your bones totally quit growing, you do have endochondral ossification happening where the long part of the bone meets the ends of the bone. So like if we zoomed in to the femur up here, there's the end of it, there's the epiphyseal plate, and there's the middle of it, the diaphysis. So it's growing right there. And the way it grows is very similar to where it grew in the first place, which was you started with cartilage. You started down here. Those cartilage cells grow, divide, then they get bigger, which is called hypertrophy. Then they get calcified, and then they turn into those osteoblasts, put the collagen and calcium salts down.
And they grow up into the shaft.
Right, and I wish there was a better picture of this, but this is the bottom. We've zoomed into the bottom of your femur. If that makes sense. So like down here, this is the part that's eventually going to touch the tibia. So this is like your knees down here, and this is the start of your femur. If that makes sense. And so it's pushing upwards. It's making new cells, pushing upwards. They divide, get bigger, and then osteoblasts turn that collagen into bone, or turn that cartilage into collagen and crystallized salts. So the plate on the other side, if you're looking down from your hip, it will be pushing downwards.
It grows like towards the part.
Yeah, so you're putting new bone in the middle of the bone. Oh, that makes sense, on the diaphysis side. It's kind of interesting, I think. And we could take a look at a movie of it happening instead of just a picture. Maybe. Uh-oh. It's very mad.
And remodeling. During childhood, bones grow in diameter or thickness via appositional growth. Long bones grow lengthwise via interstitial cartilage growth. As the bones grow and mature, they grow through continual remodeling, a process of reshaping and resizing to accommodate changing forces.
All right, let's get to there we go, bone
While
elongation.
all bones undergo bone widening, only certain bones, primarily those of the appendages, undergo interstitial lengthening. The process of bone lengthening takes place at the epiphyseal plate.
So just to remind you, now we're looking at like the top of the femur, up here by your hip would be that part.
epiphyseal plate consists of 4 distinct zones. The zone of resting cartilage consists of hyaline cartilage and is anchored to the epiphysis. The zone of proliferating cartilage organizes a supply of highly mitotic chondrocytes. As these cells divide and increase in number, the resting cartilage is pushed outward, helping to lengthen the diaphysis. In the zone of hypertrophic cartilage, chondrocytes mature and enlarge. This maturation also helps to expand lacunae and lengthen the diaphysis. The thin zone of calcified cartilage is only a few cells thick and consists of older and dying chondrocytes. The cartilage matrix near these cells has calcified. Osteoclasts break down the calcified cartilage, paving the way for osteoblasts from the diaphysis to deposit new bony matrix.
So you kind of basically saw, starting from the top, those cells start dividing the chondrocytes that made the cartilage. So then they push downwards, they get larger, they become calcified, and then the osteoblasts replace that cartilage with the bone material, and the bone gets longer. I don't know if anyone's experienced this when they were younger, any kids having a problem, but if you happen to break a bone right here, that can be really, then the bone won't keep growing in the correct way unless it's, treated and fixed properly. I don't know, that didn't happen to me or anything. Does that happen to anybody? Heard of that at all? They're worried about it. It's not all that common, but if it did, you'd end up with like your femur not growing on one side compared to the other. It would be a real problem. Yeah, it's probably more likely to damage it down there. That's where you're. I think if it's properly set and good cast right away, then it should be fine. I think it's more of an issue if like it was just sort of ignored or it wasn't set right, or maybe if you took your cast off too early and it broke again. So you can imagine the connection between her isn't all that secure to start with. Right, there's blood on both ends. Right. Any questions on that part? All right. First question. All right, long bones add to their length at the epiphyseal plate. So in this region, which type of cells proliferate, secrete cartilage, and are eventually replaced? All right, it is B, chondrocytes. So which type of cells secrete cartilage? Chondrocytes. The cell that's going to replace the cartilage with bone, that's the osteoblast, which is probably what most of you were thinking that question said, I suspect. So which cells make the bone? Is that kind of what you were thinking?
To replace with mature.
Yeah, but the key part was
Yeah.
which cells are actually making the cartilage. All right, so types of bone tissue. So we've got compact and spongy. They're basically compact. That's just like it sounds. It's very dense, of those full of crystallized mineral salts. So protection, support, and then spongy bone is sort of, it still gives you some structure, but it also gives you a good place for your red bone marrow, where you're going to make all your blood cells. And of course, the long bones, there's compact bone, and then, in the shaft, and then spongy bone at the ends. And what about flat bones? Do they have compact and spongy? They do. So they've got a compact on the outside, spongy in the middle, compact on the other side. So you do find both types of bone tissue and both types of bones. All right, so what does the compact bone tissue look like? We are going to look at it under the microscope in lab next week. And they're arranged in osteons, which is basically like a little section of a tree ring, which we'll look at in a second. The central canal is where the blood vessels come through to provide nutrients, oxygen. And then if you see this little circle around here, that's an osteon. So there's osteocytes in there, and it's mostly full of crystallized mineral salts and collagen. Just it formed right there. And here's a bigger osteon over here. So if you sliced a bone, like if you sliced my arm this way, you'd see osteons like that. So arranged sort of parallel to each other. And if you looked at spongy bone, you don't have osteons like that. Instead, you have those little thin columns of trabecula. And there's no blood vessels inside the bone. The blood vessels are in those hollow spaces between the little thin columns of bones. And that's filled with red bone marrow, which is blood vessels, and it makes your new red and white blood cells. They're around it. So I think, is there a better picture back here? Back to our flat bone. So there'd be blood vessels in these black spaces between the trabecula. So a trabecula is one of these columns. That's a trabecula. So between the trabecula are the blood vessels. Like if you had a bunch of columns on a building, there's people milling around between them. The people would be the blood vessels, and the columns would be the trabecula. Right. And then if you zoomed in, you'd see your trabecula, and then between here, all the blood vessels would be passing, and had the red bone marrow in it. All right. Oops. So compact bone is made of osteons. What is spongy bone mostly composed of? So all right, it is there it is, trabecula for spongy bone. All right, where would you find spongy bone in adults? Oh, and and we didn't talk about these words too much, most of you knew them in lab. The epiphysis is the end of the bone, of the long bones, and the diaphysis is the shaft in the middle. So epiphysis, end, diaphysis, middle. All right, nearly everyone went to E, which is correct. So in the epiphysis of the long bones, in the middle of the flat bones, that'd be where you find your spongy bone. All right, what about compact bone? Where would you find that? It is F there, C and D. So in the diaphysis and on the edges of the flat bone. So before we get going too far, why don't we do number 2 there with your group. Venn diagram on your table to compare spongy and compact. Give yourself a little space. The yellow bone marrow is actually in both. The red's only in spongy. Right. And as you get older, some of your spongy bone changes from red to yellow.
Yeah, I was gonna say, yeah.
And then your compact will just have yellow too. Store that fat for you. Just go over that one real fast. Looks like everybody had some good stuff. So hopefully you had, for similarities, that they're both found in flat bones and long bones. What else could you say? What's another similarity y'all came up with? They both have blood vessels. I think one of my slides is a little confusing. So they don't both have bone marrow. No, I know I had that on a slide, but it's because the bone marrow is not actually in the compact tissue. It's between it. So the only place you find red or yellow bone marrow is in the spongy bone. Or, yeah, let's stick with that for now. Yeah, bone marrow is in the spongy bone. Red or yellow, depending on where it is. Some differences. Spongy's made of trabecula. Compact's made of osteon. Compact is stronger. Compact's on the outsides of all the bones. Spongy's in the middle of flat bones and in the epiphysis of the long bones. So I think that those are plenty. Hardest thing to keep straight with these is we've got 2 different ways to make bones, intramembranous, endochondral. Then we've got different shapes of bones to categorize them. And then we have 2 kinds of bone tissue as well, the compact and the spongy. So I think where people usually get confused is like, maybe they'll think compact is the same as long bone or something. So you got to keep those categories of things separate in your head. You got the 2 ways to make bones, the 2 kinds of bones, and the 2 kinds of bone tissue. They all kind of interrelate a little bit. So just try to keep those straight. And we'll see you in a few days for the next chapter. We'll finish these. I knew this chapter would take a little extra. So make sure that don't let me forget to do this.